Okay, hi everyone. So you've just seen a series of presentations around some of the COTS research funded under NESP, and what I want to tell you about now is how we've leveraged this to drive really significant real-world impact in the work that I've been leading, along with Dave Westcott, around the decision support systems underpinning Australia's COTS control program. And like everyone else, I'm presenting here on behalf of a wide range of researchers and vitally also management staff and on-water crews, all of who contributed to the outcomes and impacts I'm going to talk about here. And it is really hard to overstate the impact in this case. Since NESP began, COTS management in Australia has been revolutionised and scaled up by a more than five-fold increase in government funding, and that scaled-up program has been completely redesigned as a close collaboration between NESP researchers and COTS control program managers. The research we've done has changed what is happening on water, and what is happening on water is controlling COTS in a way that we have never been able to do before. So as an impact story, this is basically as good as it gets, and this is the story that I want to tell you about now. The starting point to this story to me is one of the biggest successes of the NESP COTS IPM research program, and that's how Australia's COTS experts, and I include in that researchers, managers and on-water staff, came together to work in collaboration to drive a 6-year coordinated research effort targeted really specifically at generating impact in the way that we manage COTS on the GBR. And we targeted that impact by asking, what were the goals of COTS control for Australia and for NESP? What things would need to be done to achieve those goals? What decisions did on-water crews and managers need to make to do those things effectively? And how could NESP researchers use science to support them in making those decisions? The plan that the group came up with back in 2015, right at the start of NESP, was captured in this diagram. And over the past 6 years, we've successfully filled in each part of that diagram to underpin decision support in the COTS control program we have today. Just after the start of NESP, we implemented surveillance and control back in 2016. A little later, we showed that the control program was successfully saving coral at sites within reefs on the GBR, and we started to leverage connectivity modelling to target control at priority reefs on the GBR. And then with the expansion of the control program in 2018, we were able to take all the work that had been done within NESP around the movement of COTS and COTS biology and combine it with some existing studies about COTS density thresholds and integrate them into a new ground-up, ecologically informed control program underpinned by a decision support system. Since then, we've been able to show that this approach is working in terms of the decision points it uses, in terms of controlling COTS at entire reefs, and in terms of the regional scale strategies employed. And NESP researchers have even started asking whether we're now at a point where we can control the entire GBR or even prevent the next outbreak. So a pretty impressive body of work from a wide range of NESP researchers, and what I want to highlight now is how it all comes together to inform the decision support specifically that is driving impact for COTS control on the GBR. So let's start with the goal that we're working towards. The goal of COTS control is to protect coral and support the resilience of the GBR. To save coral in an ecologically meaningful manner, we need to target our actions to protect coral in the highest priority locations, and then when we go to those locations, we need to implement control at a level that generates an ecologically meaningful outcome, and then we need to make sure that that outcome's maintained over time. To achieve these goals, the NESP COTS decision support system is based around 3 key principles. Reefs are managed as a whole. That means that once we reduce COTS densities, they tend to stay down. Once intensive control at a priority reef begins, it continues until COTS densities are reduced so much that coral growth can outpace COTS damage. That means we achieve an ecologically meaningful outcome. And only then are control program resources redirected to a nearby priority reef. And priority reefs are the way that we make sure that effort is targeted at the highest priority locations on the GBR. To implement these slightly abstract principles on water in the real world, and drive real world impact, control program staff need to be able to answer a series of questions based on the data they're collecting as they undergo control. And that's what the NESP COTS IPM decision support system provides. The DSS is based on two parts. The main component provides the principles and structure as decision trees that lay out how to make ecologically informed decisions at each step in the process. And the second part is a tablet-based decision support system called the COTS Control Centre. Both of these were developed under NESP and are described in detail in their respective NESP reports. And this NESP research has truly driven real-world impact because the entire operations manual, Grumper operations manual for the new COTS control program is based off it, or more accurately, they were developed hand in hand. So what decisions do we inform? How do we do that? What science is it based on? And what's on the horizon? The first question we need to answer to help decision makers to implement the control program is which reefs should we target for control? And the NESP COTS research program has answered that question. Since 2018, the control program reef prioritisation has used model estimates of larval dispersal to identify reefs important to coral resilience and the spread of COTS, and then balance that with the economic importance and social importance of reefs to create a short list of 60 to 120 reefs within each region of the GBR to target control. This is based on work led by Carlo Hock and Pete Mumby at UQ and Scott Condy and his team at CSIRO. The decisions around which reefs to visit at a given point in time are guided by principles in the NESP COTS IPM decision trees, and this NESP COTS research has driven real-world impact through its incorporation in the Garumpa Reef Prioritisation Dashboard developed by Sam Matthews. And we literally used this earlier this week to identify the priority reefs that will be targeted in the coming year. So this is literally changing what is happening on water. This is determining where those vessels will go over the coming year. This is real-world impact. Moving forward, there are some exciting innovations in this space around improved larval dispersal modelling using ensembles and incorporating that into regional scale models to come up with better regional strategies. Once we know which reef we're going to, the question we face when we arrive at a reef that we've never visited before is how can we make an ecologically informed decision when we don't actually know anything about that reef yet? The first thing that the decision support system recommends is that the entire reef be mandatoed. And this is based on the science in our 2016 NESP report that showed that because outbreaking COTS tend to aggregate at certain places around the edge of reefs, but it's really hard to predict where, the best thing you can do is man-to-tow, because man-to-tow is cheap and quick and accurate enough to detect large clusters of COTS, and the control program has the biggest impact when it hits those large clusters. In our 2016 NESP report, we showed that if you were to man-to-tow and use that information to choose where to cull, you'd increase the control program efficiency by 33 to 55%. And so that's exactly what the DSS recommends. And that then guides what happens on water. Again, real world impact. Within the next year or two, this is going to be revolutionised by artificially intelligent gliders that can cover much larger areas more quickly, and perhaps some of the science that Sven was just talking about in terms of eDNA. And the NESP COTS DSS is designed to take in those new data streams as they become available and use that to improve the efficiency of management. Once you've man-to-towed, you need to decide whether you need to cull or not. The DSS uses a simple rule to answer this question based on the man-to-tow data. If no COTS or scars are seen around a reef, we don't need to cull the reef, otherwise we do. There's a lot of nuance to this simple rule, but the most important thing to know is that since the new control program started, we've had two NESP reports, the slides are in the wrong order there, that have tracked the performance of this rule and showed that it's working well. As Dave mentioned in the first presentation, it has a false negative rate of 3% and a false positive rate of 68%. And this balance is good because we really don't want false negatives, because that would mean we were failing to dive at a site that needed to be culled. but we're not too worried about false positives because we can resolve those fairly cheaply and quickly. However, we will be able to reduce that false positive rate as new monitoring and detection methods come online, and that will increase the efficiency of the control program even further in the next couple of years. Once we decide that we are going to cull a reef, we need to know which sites to start with. And again, the rule is simple, but it's based on solid science. We start with the sites that have the highest numbers of COTS because, as I said before, the control program has the biggest impact when we hit those clusters, and the faster we hit them, the more coral we save. However, actually interpreting cull data and manta-tow data and combining them to make decisions is something that's a lot easier for a computer to do than for a person to do. And so the NESP COTS Control Centre uses hybrid modelling to synthesise and process cull and manta-tow data and provide the recommended order in which to cull sites around the reef in order to cull the most COTS and save the most coral. We're already working on some refinements to this using machine learning approaches that will allow us to increase the efficiency of the control program even further. Once we start culling a site, we also need to know when to stop. This requires detailed ecological knowledge. We want to keep going back to cull COTS until the density is so low that coral can grow faster than COTS can eat it. And this work is based on the thresholds that were calculated by Russ Babcock and Eva Plagani in 2015. And after every manta-tow and cull, the Decision Support Centre estimates whether the COTS densities are below that threshold, and when all the sites at a reef are, it moves control to the next priority reef. In the very near future, that work is going to be refined under CSIP to improve those thresholds, and that could lead to significant increases in the efficiency and effectiveness of the current control program. We also need to know how frequently to cull sites, and the answer to this depends on whether the COTS we find when we revisit a site that we've previously culled were missed during the first cull or whether they immigrated in following the cull. And as part of our NESP research back in 2017, we analysed control program data and showed that sites that were dived more frequently had a higher proportion of smaller COTS, suggesting that detectability rather than immigration was the primary driver of COTS population recovery. This conclusion has since been supported by Morgan Pratchett and his team's NESP fieldwork, showing that when food is abundant, COTS don't move very much. As part of our NESP research, we modelled the optimum management strategy under these conditions and found that frequent revisitation saved the most coral. So the NESP DSS recommends that every site is dived every voyage, or roughly every 14 days. And this is significantly different to what was happening prior to NESP, where sites were revisited every 3 to 6 months at most. So this is yet another concrete real-world example of how things have changed on water as a result of NESP research and how it's driving improved control of COTS on the GBR. After all of that, the final question is the control program working? Is it working to control COTS at sites? Is it protecting reefs? And are the regional strategies we're using effective? As a result of these 3 NESP reports, we can now say that the answer to all of these 3 questions is yes. Dave talked through some of the detail earlier, but just to reiterate quickly, our early NESP results showed that even under the old control program, coral was being saved at individual sites that were being visited frequently, but no entire reef was being protected. The major innovation and impact under NESP has been that the new control program that we designed is now managing COTS across entire reefs. It has reduced COTS densities to a point that coral can grow faster than COTS can eat it at 89 entire reefs, as well as verifying that COTS are below that threshold at an additional 99 reefs. So that's almost 190 reefs that we can confidently say coral is growing faster than COTS is damaging it, that we couldn't have done without this NESP research. And finally, under NESP, we have used 2 GBR scale models of COTS control, CocoNet at CSIRO and ReefMod GBR at UQ, to show that the regional scale strategies that we're employing are optimal. And so not only have we shown that we have achieved the goals for COTS management set at the start of NESP, but NESP researchers have just started publishing a couple of papers showing that we're right on the cusp of being able to control COTS right across the GBR or even prevent future outbreaks entirely. So we're not there yet, but we're so close. And that's really exciting. So that's how the NESP COTS decision support system we've developed with on-water managers to underpin Australia's COTS IPM control program delivers real-world impact by taking all the science funded under NESP and translating it to guide on-water decisions that generate the most number of COTS culled to save the most amount of coral in the most important locations on the GBR. Thank you very much.